Welcome to Careers in Discovery, your window into the world of leaders in pharma and biotech. Brought to you by Singular Talent, making hiring better for organizations involved in drug discovery and R&D. Ali Tavassoli is the Chief Scientific Officer at Curve Therapeutics, a company developing a revolutionary intracellular screening platform to power drug discovery for challenging targets. Ali joined us to talk about his career balancing academic work with building a biotech company, resisting the pressure to publish when tackling major scientific challenges, and why following the science is crucial. This week on Careers in Discovery, I'm delighted to be joined by Ali Tavassoli of Curve Therapeutics. Ali, welcome to the show.
Hi, Tom. Nice to be here.
Great to see you. So I'm really intrigued, Ali, to learn a bit more about Curve and your screening platform, the work that you're doing there, and how it all came about. So tell us about that to start with.
Yeah. So the screening platform that Curve used, it originated actually from a time when I was a postdoc at Penn State.
Mm-hmm.
So this was about 2003. So I initially moved to Penn State to work on a synthetic chemistry project. I was a synthetic chemist by training.
Yeah.
We made a few compounds. There were methyltransferase inhibitors. They ended up being quite good antibacterials and antifungals. That was spun out into a separate company. And actually those compounds are one of them's on the market called Tavassoli. Yeah, it's an antifungal. So after that, sort of I didn't have much to do, And I went to speak to my then supervisor, Steve Benkovic. And I said to him, maybe I should just head back to Britain. Maybe I should just go and look for a job. I think I'm done here. I've done my bit. And he's like, no, Ali, why don't you hang around? And I was like, well, what do you mean hang around? He's like, you're a smart guy. You'll figure out what to do. Just hang around. Are you going to still pay me? He's like, yeah, just hang around. So I was in this really fortunate situation where I could just talk to my colleagues, fellow postdocs, and that was a wonderful lab to be in. It was a very multidisciplinary environment. There was chemists there was biologists there was biophysicists. So it was a really good environment to be in to try and come up with a new idea. So we talked and we talked and we talked. And there was this technology that, a couple of postdocs, like 2-3 years before I was there, had developed in Steve's lab, which used these protein domains called inteins to cyclize things. So you could put GFP between these two protein domains, and the domains would come together and they would cyclize whatever you put in the middle. And it's a proper head-tail cyclic, amide bond. So we sort of thought about this, and I was really interested in what used to be called undruggable targets in those days, sort of your protein-protein interactions. And we sort of started thinking about, me and 3 other postdocs, we started thinking about, how do we start tackling this? And we thought about using Cyclops as a means to generate libraries, because if you have a domain that will cyclize whatever you put in the middle, a genetically encoded domain, you can randomize at the DNA level. That will give you various products, right? And if you only put 6 amino acids in between these two domains, you get a cyclic hexapeptide. And with, the position 1 needs to be set, a serine or a cysteine for the splicing process, but otherwise you could have any of the 20 amino acids. And, if you have 5 random positions, that's a library of 3.2 million entities. And these are being made inside cells. If you think of traditional drug discovery, most of drug discovery happens in a biochemical buffer, FP assays, biochemical assays. So if you want to do high throughput screening, by definition, the target you're screening against is not in its native environment.
Right. Yes.
It's in a biochemical buffer in a single conformation. It's not in a cell sampling multiple conformations interacting with its natural partners. And so this ability to make a genetically encoded library inside cells was particularly exciting. At that point, we coupled the library to a bacterial reverse to hybrid system. So we would engineer bacteria so that their life and death depended on the interaction of 2 mammalian proteins.
Right.
And then if a member of the library broke that PPI apart, protein-protein interaction apart, the bacteria would live, we'd isolate it, we'd sequence the plasmid, that coded for the cyclic peptide, and that was your inhibitor. The thing that, Steve was kind enough to let me bring that with me when I came to Southampton in 2006. And I've been working, me and my lab, my team have been working on that technology, improving it ever since. The real step change that allowed Curve to be born were two things. One was we figured out how to do this in mammalian cells.
Right.
So, we can generate the library and do the screening inside mammalian cells.
Yeah, I see.
And we've coupled this to next-gen sequencing, and that allows us to have really high-quality data sets that we can then go and interrogate for, what is the best inhibitor to take forwards. And then the other platform we developed was a microfluidics-based platform where you can generate the library inside femtoliter water in oil and water double emulsion droplets. And again, similar to the mammalian cell screen, you can use next-gen sequencing to identify hits. We have data mining tools, programs that will go and look at these sequences.
Yeah.
Because the hexamers are special. The hexamers aren't there's several other techniques that allow you to screen cyclic peptide libraries. Phage display can look at cyclic peptide libraries. There's a technique called mRNA display, which can also be used to screen cyclic peptide libraries. But in those assays, and DNA encoded libraries suffer from the same shortcoming.
Yes.
You are screening in biochemical buffer on a solid surface for binding. You're not screening for function, you're screening for binding. And so what you get are relatively tight binding molecules that don't necessarily function.
Right, yes.
When they go in a cell.
Yeah.
And sort of what the Curve technology overcomes is that very fact, because you're now screening for function, and
Yes.
you're doing this inside a cell with a massive genetically encoded library. And the hexamers that we use, these are much smaller than the cyclic peptides used in phage display and mRNA display. They're much more rigid, and what this allows us to do is, studying them over the years, I found that actually they interact with the target via 2 or 3 amino acids within that hexamer because of the physical properties of where the side chains point in a hexameric cyclic peptide.
Yeah, I see.
And so we've managed to scaffold hop out of a cyclic peptide into small molecule backbones. So, that's a really big thing for us as well, because, the whole field, the cyclic peptide libraries, you're presented with a problem that the cyclic peptide is your drug. And the output of an mRNA display, phage display library, those peptides are big, 18 MERS, 20 MERS. And they need to be, because you're relying on that binding and the slow off rate in the assay, right? You put the library on a target, DELs are exactly the same. You put the library on a target, incubate a bit, wash to wash off things that aren't binding tightly enough. And what you have left, you can sequence to see what the entity was. And so these need to be fairly big, lots of hydrogen bond contacts to stick. The hexamers don't suffer from this because we're not screening them in the same way. They're being screened inside a cell. And because that pharmacophore is 2 or 3 amino acids, that can be very, in a fairly straightforward way, scaffold hopped out of a peptidic backbone into a small molecule backbone. And so your path to the clinic is that traditional medicinal chemistry, well understood path.
Yes, I see.
Of course, it has its pitfalls, but it's far better understood than trying to take a cyclic peptide to the clinic, which has been challenging for most companies that have engaged with it.
Yeah, absolutely. So there's several ways that it does this, but essentially your platform is making these hits more translatable, right? More likely to succeed.
Indeed, because at the screening stage, they're screened in the relevant cells, relevant disease relevant cells. And you could do really interesting things, right? So for example, you can take isogenic pairs of cells and look for synthetic lethality. And just a blind screen, what kills these cells but doesn't kill the isogenic pair. And because we have the next gen sequencing tools and the data analysis tools, you sort of zero in on the pharmacophores sort of relevant. It clusters the hits because multiple peptides would contain that, say, tripeptide pharmacophore.
Yeah.
But the activities will be slightly different depending on the orientation in which the other side chains present the pharmacophore. And so we get these little clusters of hits, around the pharmacophore, and you can go and make any of those and they'll be active to some degree.
Wow. Okay.
It's really is very interesting,
Yeah, fascinating. And so, as you said, this came out of some research that you're doing in your postdoc, and we'll come back to that. Nice to have the sort of freedom that you had there as well to come to hit on this idea. But you've stayed with it, I guess, for some time after that, and there's been a lot of work gone into it, of course, on the back of that. And so Curve has come out of that research. I know you also still spend time at the University of Southampton, and you're a professor there.
Yes.
So tell us a bit about, I suppose, starting the company, and you said there was a couple of breakthroughs that triggered that, and also I'm really interested in how you manage your time around these two jobs that you have, Ali, because I imagine you wouldn't describe either of them as part-time.
I mean, it's interesting because I do get paid in the part-time for both jobs. I'm only at 25% of the university.
Yeah.
But you sort of think, okay, which bit of the job am I not doing? So it's, both university and Curve on part-time nominally, but you have to. And it's an exciting time, right? It's a good kind of busy. There's bad busy where you're spending your time doing things you don't want to be doing.
True.
And you don't see the point of why you're doing, why they're making you fill a format. You're like, why are you making me do this? But there's also the good busy where, the things you've imagined are coming true, things you've designed are happening and they're working, on multiple fronts. And that is a good busy. That is something that you don't mind spending
True.
your evenings and weekends on. It's really great. I mean, I've got a young family as well, so that balancing the time and making sure the kids get to spend enough time with me, and my wife sees enough of me, and that's all a balance. That's all a balance. And obviously, despite best efforts, it's something that you're never going to get perfectly right.
No.
But I mean, I felt when the opportunity came to start Curve, and it was serendipity, really. I met our current CEO, Simon Carey. I met him maybe 6 years ago now. I was looking to start Curve. I went up to his previous company because it was a Southampton startup. And I went to his previous company, actually someone from our research innovation services, James Hamilton, he took me up there to talk to Simon for like a couple of hours.
Right.
And Simon got it instantly. As soon as I said, look, this is what the tech can do, he just got it instantly because he has a background in this sort of thing.
Yeah.
And he sort of helped me a bit. He was like, I can't get involved right now because I have a job, but let's keep talking. And he helped me with a bit. And then a couple of years later, I got an email saying, look, I'm now part of VC. I'm part of Apple Life Sciences. I'm a partner there. I think you should come and pitch this idea to us. And it was, completely, just a complete surprise. And I went up. We went up and pitched the last day of Christmas. The last day before the Christmas closure.
Yeah.
And the term sheet was back from the VC, early January. So it was something that they could also see the potential.
Okay.
And this is why it's been so wonderful, because when other people can see the potential,
yes,
that you can, I think it really drives you on. At the time that the people investing did require that I join the company as CSO, because obviously the technologies come out of my lab and I know probably the most about it. So it wouldn't be fair on the investors if I didn't give them a full commitment to make sure that the company succeeds. And I really wanted to as well. I'd hit a stage in my career where, I was a full professor.
Yeah.
I had them for a few years. I kind of wanted a change,
Yeah, I understand.
But wanted a change in circumstances just to keep myself, keep on learning things.
Yes.
And keep on doing new things. And so it came at perfectly, the perfect time, really. It came at exactly the right time. And I didn't really ever think about leaving the university because I do really enjoy teaching. I do like, I think it's a responsibility for me to pass that excitement for chemical biology and that interdisciplinary science, that working at the interface of chemistry and biology, pass that down to the undergraduates that are coming through.
Yeah, makes sense.
So, I have one course that I teach, and I felt pretty passionately that I want to keep doing that. I have my research group,
Hmm.
and again, that allows me to do the crazy stuff. That really is a good vehicle
Yeah.
to develop the really interesting, crazy ideas. And then I think the group are very good. The group are excellent. My academic group, I have senior people who are just brilliant, and they really look after the junior people coming through.
Hmm.
And the junior people are engaged, and they really, smart people that they understand what we're trying to do, and they understand the importance of what we're trying to do.
Yeah.
And so, the group is an absolute pleasure, as are the scientists at Curve. I think,
Mhm.
again, the whole team, we're still small enough that, I know everyone, I see everyone on a day-to-day
Yes.
basis. And you sort of really see that they get it. They know why this is important, and they're pushing hard because they passionately believe
Mm.
science that they're doing. And, it's just brilliant.
Yeah, absolutely. No, it sounds great. And in terms of balance, you just got to get the kids up to speed in the lab, and then you can see them all the time, right?
Yeah, yeah. I did try, but I think the university wasn't happy with a 6-year-old doing lab work.
Well, that seems unreasonable to me, but
It does, right? Doesn't it?
So we've talked a little bit about your earlier career as well, Ali, and we'll go back to that. But we always like to start at the very beginning. And for you personally, why science in the first place? Why chemistry, chemical biology? Why this career for you?
Yeah, I mean, there is a parallel universe in which I end up being a dentist. I
Hmm.
won't lie to you. So, I come from a family of scientists. So,
Yeah.
my mum's a chemist, my dad's a chemical engineer.
Okay.
So even the generation before that, they're scientists and engineers. So I come from a family where science was just there when we grew up, and sort of for me, I always found science most interesting. Science and mathematics I always really, really enjoyed
Yeah.
as I was going through school. And so A-level choice, I did chemistry, biology, and physics. And I always kind of wanted to be a doctor, but my grades were never ever the top that you need to be a doctor
Right.
in the day. And so I thought, well, maybe I'll be a dentist. So I applied for dentistry, and I got into like a few dental schools. And the summer before I was due to go to university, I arranged to do work experience for a couple of days at our local dentist. And I saw wisdom teeth extracted.
Right.
I don't want to do this.
Yeah.
was so much blood. So I just like, oh, yeah, no, this isn't for me. So I called up the universities where I've been accepted into dental school, and I sort of asked them what else I could do.
Yeah.
And Bristol University said, okay, you can switch and pick one of your A-level subjects to switch into. And I enjoyed chemistry most, so I thought, okay, can I get into chemistry? And they're like, yeah, sure, come and do chemistry.
Yes.
So I kind of feel my whole life has been a series of accidentally taking or A or B, and it just I guess most people's lives are this way.
Yeah, I think so. It comes up a lot more often on this podcast than you might expect.
And I sort of did my degree, and then I really enjoyed it, and I knew I wanted to do a PhD. So I did my third-year project in inorganic chemistry, developing platinum catalysts. And I thought, well, no, I do want to be a bit closer to drug discovery. I was always passionate about developing new drugs.
Right.
I was really passionate about using chemistry for a purpose, and the purpose I wanted to use it for was to help people who were sick.
Yeah.
And so I wanted to do some medicinal chemistry or synthetic chemistry. So I asked around, and one of the professors there, Joe Sweeney, he said, "Yeah, come do a PhD with me." And he had what's called a CASE award in the UK, so a company sponsored award. And so I had a PhD offer to do a CASE PhD with GSK.
Right.
And so I accepted that, and sort of a couple of days, not quite that bad, maybe a couple of weeks before we were due to start, he called me up and he's like, I got some news. I'm moving to Reading. So he was in Bristol at the time. Oh, really? So like, fine. So we found a place to live in Reading, and, you know, all of us, there was 3 of us who started that year. And we all sort of went into the lab in Reading and started setting stuff up.
Yeah.
So that was a really great experience to, you know, setting up a new lab and starting.
Absolutely.
And the project was just methodology development. So I spent most of my PhD doing the same reaction under slightly different conditions and it not working. and you know, 6 months before the end, it worked. 6 months before the end, I hit on the conditions. And so I was working day and night to get enough data for my thesis, so to get a PhD. And actually it was all good in the end. You know, my viva lasted about 30 to 40 minutes, I think. It was really quick. Yeah, it was really quick. I was, you know, because normally they're meant to last about 3 hours, and we had bets and in the group how long each viva would last, and the sort of start time was an hour and a half.
Right.
I think the examiner just came in and looked at it, and he asked me a few questions, and I always answered with far too much detail. You know, PK, this is who the reaction was invented by. It's like, you know what you're talking about. It's all good. Let's all just go to lunch. You're fine. I was like, oh, okay.
Oh, good. Well, at least they told you in the meeting rather than 30 minutes, and then you had to.
So it was really quick. It was just such an incredible. And then so after that, I wanted to learn a bit more biology. And I wanted to go to the US. You know, I tried really hard after my PhD to go to the US. And in those days, we didn't have email, right? There was 99. My email was in its infancy, so you still had to write letters. So, you know, in 98, 99, I must have sent about 50 odd letters to labs in America. And I'd get replies back, but they would all say the same thing, which is, you know, you're welcome to come, but you got to work for free and you got to have your own money.
Right.
So that wasn't obviously an option for me. And so but I did want to learn biology. So I took a postdoc at Sussex with Doug Young, who was one of the pioneers of the bioorganic chemistry field.
Right.
And Sussex was a real hotbed of science at that point, Sussex chemistry. You know, there was 2 Nobel Prize winners there, and it was just a really excellent department. And I mean, it still is. And so I really enjoyed my postdoc with Doug. You know, I was the senior postdoc there. I was, you know, really getting the group excited and interested about chemistry. You know, we had things like PK of the day, where I put a structure up on the blackboard, and I'd ask them, you know, what the PKA was, grill the students. We had reaction of the week, where I'd teach them a name reaction. You know, it really showed me how much I enjoy teaching.
Yes.
And, you know, teaching things to students. And so after that, Doug knew Steve pretty well. And sort of towards the end of my time there with Doug, and I think Doug was, he thought of me highly. And so he said, look, Ali, if you want, I can call Steve. I know Steve. I can call him, and I can get you a postdoc in the US if you want to go to the US. But it's like, we have to promise me that you're not going to mess Steve about. So if you accept the postdoc, you've got to go. You can't go for other interviews and just turn them down, because, you know, I'm going to put myself on the line here. So I said, yeah, absolutely. I mean, Steve's huge. You know, Steve is one of the inventors of the chemical biology field. You know, he is so well known. Enzymology, the work he's done with catalytic antibodies, the work at that point with the replosome. He's really was one of those big names I've always admired. And it was just incredibly easy, you know, compared to sending 50 letters. Doug, you know, calls me up to his office one day. Steve's on the other end of the phone. Steve talks to me for about 5 minutes, asking what I've done, and then says, okay, do you want to come? I was like, yeah. So okay, see you in September. And then puts the phone down. And it's just, wow. You know, and I think when you're meant to be somewhere, that's how easy it can be. And if you're trying to swim against the tide, you can try. But if you're not meant to be somewhere, you're not going to be there.
No.
So that, or maybe that's just me justifying letters, you know. So
sometimes I think you're right. I think sometimes these things just come together, don't they?
They do. And so I went to Penn State, And I think I talked about it at the start of speaking to you. It was just the most incredible environment, you know. It's just incredible. There was 20 odd postdocs there.
Yeah.
All different in training, and all coming together, pulling together. And it really opened my eyes to what is possible with big science, you know, where you have a large problem that you work on as a team.
Yes.
And you know, I think in some ways that the funding model in the US allows that a bit better than the
I was
funding.
going to say, because you don't always find that in academia, do you? You don't always find that people are pulling together and working together.
It's unusual. It's unusual. I think, yeah, it is. And partially it's the way we fund science here versus the way science is funded in the US. Partially it's just that can-do attitude that they have over there. I think just the attitudes are slightly different. We're slightly more cautious here.
Yeah.
A bit more gung-ho over there. So but it is different. And, you know, that's one thing I always recommend to my students and postdocs is go and sample another country's science. Go and sample, you know, and yes, it can be scary and it can be tough. And it is tough, you know, you're leaving behind your family, you know, you're going into a whole new environment that you don't really understand. But the clarity you have of very little distractions working on a problem, right? And you sort of day and night you're working, but you're working because you love it. You're not working because someone have to work this hard. You're just like, I want to solve this problem.
Yes, yeah.
You know, and the sort of key bit of data that came through. So, you know, when I had switched over to the Cyclops project and we were building the bacterial 2 hybrid systems and all this stuff, the key killer bit of data that came through, you know, because you build these bacterial strains and I wasn't a molecular biologist, so it took me forever.
Right.
You know, and you do the screen and then you've got your peptides, you have to make the peptides, you have to express the proteins. The enzymatic assay, does your protein, is your protein inhibited by this peptide that you've spent all this time building a bacterial strain for? That bit of data came in at about 10:30 p.m. on a Friday night.
Right.
And I was literally not quite in tears, but close to tears with the happiness. You know? There was just no one to share it with. I was running around the lab to see if anyone's there to tell them this had worked. And I was like, I'm going to have to wait till Monday to tell people, you know, this is crazy. So it's that sort of thing where you're just driven by the science, by the problem, by solving the problem. You're driven because you want to solve the problem. I think, you know, you try and think your way through the issues that come up, you know. I think that's what I really, really, really enjoy. And I think once we had that bit of data that the system worked and everything worked, and again, this wasn't just me, right? There was
of course.
3 of us, 3 others. There was 4 of us on the project, you know, Alex Horsfield, Sergei Sabanov, Todd Nauman. You know, we worked together as a team. And as again, we all had slightly different skills, but we all really pulled together. And it was just such a wonderful environment.
Yes.
You know? So yeah, I really think back at my time at Penn State and think, you know, really fondly look back on it.
No, it sounds like it. It sounds like it. And then you talked a little bit about what happened there with that project and the other research that you did, and then you headed back to Southampton and I guess started doing some more teaching and started working on this problem further, on this screening platform further.
Yeah, so the first target I picked was hypoxia-inducible factor. You know, this is a protein that senses oxygen levels in cells and responds as soon as oxygen levels drop. And it does this by changing transcription of hundreds of genes. You know, that cell needs to fundamentally change in order to survive in a hypoxic microenvironment. And this is important for fetus development, you know, because the blood vessels need to develop, so new vascularization. And so signals are sent for that, but also the metabolism of the cell changes. You obviously can't have the Krebs cycle anymore, so the cell becomes glycolytic. It sends out signals for angiogenesis, you know. So it's a really important pathway for development of a fetus, but it's also critical for cancer cells.
Right.
Discovered in 95 by Greg Semenza, and it was pretty obvious. There's 300 to 500 papers a year on HIF every year since then, and it's a really critical protein in solid tumors.
Mm-hmm.
And so and a lot of companies have tried to get inhibitors, because it's a transcription factor.
Right.
It's made up of alpha and a beta subunit. So being interested in protein-protein interactions, I wanted a compound that broke that alpha and beta subunit apart, and so stop the transcription from assembling.
Mm-hmm.
The way the protein works is the alpha subunit is constantly made and constantly degraded in cells. So it has a half-life of about 5 minutes. This is how important this pathway is. Every cell in your body is spending valuable resource making a protein that it degrades 5 minutes later.
Wow.
But if the cell becomes hypoxic, that degradation pathway doesn't happen anymore because it uses oxygen as a substrate for the enzymatic hydroxylation. And so alpha builds, and alpha gets transported to the nucleus where beta is waiting.
Right.
And they dimerize and change transcription, allowing that cell to survive. So if you could prevent that protein-protein interaction from taking place, you would stop cancer cells from being able to respond to the hypoxic microenvironment.
I see, yeah.
And it's been hypothesized that it would have a significant therapeutic effect in cancer patients. So I, when I first started my own lab, wanted to find an inhibitor.
Yes.
Some would say quite a foolish thing to do, right? It's quite a difficult thing to do, because when you first start a lab, you don't have a massive amount of resource. You don't have a lot of people. And so typically people ramp up how hard the problems they were. I went straight for that.
Yeah, good.
It was quite challenging because you get pressure to publish. You get every year you're being reviewed, and the person reviewing said, "Well, where are the papers, Ali?" And I was really busy writing grants, and I was very lucky that I was really well supported by the funding agencies, and by obviously my colleagues reviewing my grants were very kind to me, and could see the importance of what we were trying to do, and were saying that. So I got a sort of fairly sizable group straight away, fairly quickly, but we were working on a big problem, and big problems take time.
Yes,
and
of course.
To have that, I don't know, self-belief, I don't know what you'd call it, where you get pressure saying, well, why aren't you publishing? Where are the papers? It's been 4 years. Where are these big papers? And to have that self-belief to go, no, you need to wait. We're working on a problem. This needs to be right when it goes out. And I think that's always been a feature of my of my characteristics, where I want things done right. And if there needs to be I'd rather produce a few high quality items than lots of less important things.
Yeah, understand.
And it's not better or worse. I'm not saying it's better than the other way. It's just how I am. And there's advantages to that, and there's disadvantages to that for me, So, but initially there was to resist that pressure to publish, and then finally we published the paper, and even now it's still the only isoform specific. So it hits HIF1 over HIF2, so it's isoform specific.
Okay.
Inhibitor, which sort of speaks to the importance of screening inside cells for function.
Right.
HIF is so important. Most pharma companies have screened for HIF. They've tried to find HIF inhibitors without success. And so, we've gone back and we've worked on dual inhibitors of HIF1 and HIF2. And there's a compound called belzutifan, which was developed by a startup called Peloton from Texas, and bought out by Merck. And that inhibits the dimerization of HIF2 alpha, which is the isoform with HIF1 beta. And that works, really great. The issue is only a small subset of cancers are driven by HIF2. So it's only renal cell carcinomas, and only a subset of renal cell carcinomas that are driven by HIF2. It works in the clinical trials. I think the partial response was the response was 50% of people responded,
Yeah.
53 or something, and they were all partial responses.
Yeah.
So what you really need is a dual HIF1, HIF2 inhibitor, something that breaks apart HIF1 alpha dimerizing with HIF1 beta and HIF2 alpha. And that's what we've developed. We initially started working on that in the group, and then the compounds were exclusively licensed to Curve when Curve started up, and they've successfully converted those to small molecules. And we're just in the process of sort of developing them into leads, and then hopefully in a couple of years we'll be starting trials.
Yeah, fantastic. And I suppose thinking about Curve, I guess you're 3.5 years into.
Yeah, we're 3.5 years old now.
3.5 years, yeah. Has it been what you expected? What have you learned through going into a biotech company and spinning that out as opposed to an academic lab? And tell us a bit about that period of time.
It's been wonderful, because I was getting, not bored of academia is the wrong way of saying it, but I wanted a change, and it's been like moving to a new discipline.
Right.
It's so different. It is so different. And I've been lucky in that I've got a very experienced CEO, Simon.
Yes.
Who's been guiding me through this, I can't imagine doing it by myself,
No, of course.
And I think that's been really, really good to have Simon here, and he teaches me things about the business side of things, and it's been really great, and it's really turbocharged our science. The sort of biggest thing that it's done, the company, it's completely turbocharged. And I was saying this to someone the other day, that if I had not gone down the Curve route and I'd just carried on as an academic, it would have probably taken me to the end of my career.
Right,
I've probably got another 20 years left in academia. It would have taken me to the end of my career to get to where we are today with Curve in 3 years.
I see, okay.
It's just incredible, because essentially there's 15 postdocs out there working together with no red tape. If I want an instrument, I don't need to fill in a massive form. I don't need to justify it. I don't need to write a grant, wait a year for that instrument to get the grant with like a 20% success rate. Then don't get it, then write again. Maybe you get the instrument, maybe the university can't pay half of the cost of the instrument, so you don't get, you just can't buy it. And get the science done. And it is so refreshing to have the science come first.
Right.
I think universities and the funding model has changed over the past even 10 years, right?
Yes.
We've got a lot more admin, we've got a lot more red tape. And not just universities, the funding agencies are just as responsible for this. ARI, they're just as responsible. Everything now needs a form, everything needs a million justifications.
Yes.
And science almost takes a back seat. And just the nature of the funding is that, ideas that are a bit higher risk and out there, they don't always get backed, because, it's just how it is with the way things are run at the moment.
Yeah.
And so, it would have easily taken me to the end of my career. And maybe we wouldn't have even got done half the stuff we've done at this point at Curve. So it's been really great, and it continues to be really great. We're going through a really exciting phase at the moment, because, when we started, a lot of the things that we wanted to do were just ideas.
Right, yeah.
And a lot of the tech has had to be developed on the fly. I think someone said to me, it's like being thrown out of a plane with some material that you need to make into a parachute before you hit the ground. When you've made, I'm sure that analogy has been used lots, but when you successfully make that parachute, and with Curve, it's on 3 different fronts, right? So we've got the challenge of converting the cyclic peptide to a small molecule, scaffold hopping. That's been done successfully. We've had to set up the mammalian cell-based screen, set up the data analysis software, set up the tools we need to do that. That's all been done, and we're getting hits that we can validate now that are functional in cells and in vitro. And the microfluidics platform has had to be set up and run. And again, that's running fine, and we get hits that we can validate on that. So it's just touch wood, everything's working on all fronts, and it's just very, very sort of satisfying.
And that's exactly where you want to be, right? And it takes a lot of work to get there, but it's nice when it starts to all come together.
It does, and I'm sort of lucky that my wife's a scientist also, so she's sort of very supportive, and she understands that.
Yes.
Sometimes evenings and weekends are needed to get stuff finished.
That's helpful. And so, Ali, I'm asking you to summarize quite a significant period of time here, but if you look back on your career to date, and I suppose if you were talking to someone who was maybe just coming out of their first postdoc, or was thinking about taking a step into a biotech company, or it was early in their career as a scientist, what are the sort of one or two things do you think you've learned from a career point of view that you'd share with that person, or that you wish you knew when you were starting out?
I think there's two key things really. One is always go with the science. I know a lot of, if you don't believe in the science, and as someone joining a biotech, you should always question whether the science is good or not, because there's lots of biotechs, and there's the old hype cycle where everyone's working in one area, and you need to make sure that actually the science that you're going to spend your time on is good. And if you believe in the science, then all will be fine. I think that's the biggest piece of advice. And then the sort of second, and I've always done that, without maybe fully knowing it. I've always done that. I've always had multiple paths open up to me that, you can go this way or you can go this way. Even at the time when we started Curve, there was 2 or 3 other options. And I sort of thought, no, I believe in this. I'm going to back myself. I'm going to back myself, and I'm going to push on this,
Yes.
And that's the second piece of advice, is really back yourself. If you don't have belief in your own abilities, how can you expect someone else to? And I think there's nothing worse than a hollow belief, right? So you need to be honest with yourself. And if you're no good at something, be honest with yourself.
Yes.
But if you do genuinely believe that this is right and this is the way to do something, then back yourself. Back yourself and go with it. I think that's really the key thing for me, is because I could have stayed in academia fully. I could have carried on being an academic till I retire, and I probably would have had a bit of a easier time of it, in terms of hours I used to work and the stress levels and things, But it would have been a lot less interesting and a lot less exciting. And there's a lot of things that just wouldn't have happened. It wouldn't be out there in the world if I'd done that. So it's been that belief that you should always back yourself and back your thoughts and back your ideas is what I really recommend to people early on. And really have an idea. I'm not one of these people who says have a 10-year plan or a 5-year plan. I've never done that. I've never gone, 5 years time I want to be there. I'm going to picture it, therefore it's going to happen. I've never done stuff like that, but I've always had a rough idea of what I want to do and where I want to be.
Yeah, I understand.
And everything I've done, I've made sure is a step towards that. And the science, especially when you're going into a biotech company or you're going doing a postdoc, that science stays with you forever, right?
Yes.
You work on that protein and it's part of you now, or you work on this inhibitor and you understand this pathway. When you move on to your next job, you take a piece of that with you in your head, and so you want to really make sure that these things become additive. That I want to be here, therefore I'm going to do a bit of this, I'm going to learn a bit of this, then I'm going to do a bit of that, and that's. I think that there is,
doing that with some direction helps you to bring it together, right? It's if you make those decisions on their own merits, you end up often somewhere that you didn't expect to be.
Exactly.
And you mentioned for Curve, it's an exciting time. The technology is really coming together. Things are working. Tell us a little bit about what's next and what the next phase is for you guys.
So we're just in the process of raising some more money to allow us to grow the team. So the team is going to grow to about 60 people. We're currently building or renovating labs on the Southampton Science Park, so we should be in our new home just before Christmas. And again, that's been wonderful to be able to sort of design the labs, and this is where we want this to, how we want this to be, and so that's really an exciting time for the whole team. And once we raise the extra money, we're gonna point the HIF project towards the clinic and start working towards getting that in that direction. So that's going to be incredibly exciting. I've been working on HIF all my career, and to sort of get the fruits of that labor in such a meaningful way will be just wonderful. It will just be wonderful.
Absolutely.
And then the screening platforms that we've developed, those are obviously, we have internal targets, we have internal programs. We've also very recently this year announced a sort of collaboration agreement with Merck, US Merck. And so they have the option of nominating up to 5 targets. We're currently working on 2 targets with them, which again is really great. They're such a great company, such a great bunch of people to work with, and so it's really interesting times. I think I am going to have to make a decision about just reducing my academic footprint just a little bit, just because as Curve grows, and I'm enjoying, being part of that so much, that I just don't think I'm going to have as much time to dedicate to the academic side of things. For a while. So
I think that's a common situation to find yourself in, and there's only so much time, as you say, and you don't tend to get less busy as companies grow. Well, it is an exciting time, Ali, and as you say, seeing all that work that you've done heading towards patients, I'm sure will be very exciting as well, and we wish you the best of luck with it.
Thank you so much.
Thanks for joining us on Careers in Discovery, and don't forget to subscribe for more insight into the world of drug discovery and R&D. Do take a look at our sponsors, Singular Talent, and their mission to make hiring better for companies and individuals in drug discovery and R&D. You can find them at www.singulartalent.io. See you next time.